Yeah, we're okay. Welcome to the afternoon session, and the first speaker is remote. He is Luca Turin, who's very well known in the field of quantum biology, and particularly of olfaction. He's quite famous for that, and he's done some very interesting work more recently with spin. And he was going to be here, but I think his wife had surgery, and I hope she's okay, and I hope he's available. So, Luca, are you there?
Yes, sir.
Hey.
I'm here.
Good to see you. I hope everything went well.
Good to see you, Stuart.
We miss you.
Yeah, I'm sorry I couldn't come.
Okay. I can't remember the exact title of your talk, but it's about Spintronics and...
Yeah, Spintronics in the brain.
Okay, here you go. Luca Turin.
All right. Thank you so much. Thank you. Well, let me start by thanking Stuart and Thomas Brophy for inviting me to this. It sounds like an absolutely fantastic conference. I'm really sorry I couldn't make it. But here I am, 10 p.m. in London. I think it's the latest lecture I've ever given. And let me start by emphasizing how important Stuart's direction has been over the last few years, in my opinion, in the field of neuroscience. Stuart has brought to everyone's attention repeatedly and insistently the fact that anesthesia is probably the best gateway to understanding consciousness. The fact is, we have a tool to turn off consciousness reversibly. And as Stuart has pointed out many times, the anesthetics work on just about every organism that we can detect being asleep. And so it's more than likely that it acts on a fundamental cellular mechanism or subcellular mechanism, which we absolutely must understand before we can go forward with understanding consciousness. And I think Stuart's been very much at the center of this idea and keeping everybody's minds focused on this. Let me now share my screen and show you what I'm going to be talking about. So I'll be talking about consciousness via anesthesia. So general anesthesia started pretty much by accident in the 1840s. There's some dispute as to where and when, but roughly speaking, 1846 is a landmark date. And this poor man was anesthetized and the neck tumor was removed. And it's pretty remarkable that 177 years later, things are fairly similar. I mean, nobody uses ether anymore. They use halogenated ethers because they're not flammable. The patient monitoring has become much better, but basically it's the same thing. And in a tight spot, you could still do a perfectly good ether anesthesia following the directions of a little book you can buy. And you could probably put someone to sleep for half an hour without too much risk. Now, the thing about anesthetics is that they are the strangest category of drugs in all of pharmacology. This slide shows a few of them, roughly ordered in increasing size. And, you know, if you have any knowledge of pharmacology, this will cause you to despair because there is no visible structure-activity relation. And if you see one, please drop me an email, because as a matter of fact, the fact that xenon, which is a sphere of electrons roughly 4 angstroms in diameter, and a steroid like alfaxanone are both anesthetics is really quite extraordinary. And 177 years later, we're not very much wiser as to exactly how these work. Now, I just want to show you xenon. Xenon, by the way, was expensive and hard to get, but now it's expensive and impossible to get because almost all of it was produced in Russia, and the Russians aren't selling it at the moment. But look at this wasp. Oops, the video. For some reason, the video didn't work. Anyway, the wasp on the left was totally knocked out by xenon instantly, and the wasp on the right is the same wasp 20 minutes later, and it wakes up. This is amazing, and xenon works on humans just the same way. It's not used because of the expense, but it would be a perfectly good anesthetic. So, what's so weird about anesthetics? Well, if you think about pharmacology, the way you make proper molecules, not weird gases like anesthetic gases, stick to a protein is via the hydrogen bonds they make or some other interactions. And for example, biotin and avidin, it'll stick to it. Biotin is a small molecule that sticks to the protein called avidin essentially forever because it makes so many hydrogen bonds that the off rate is absolutely tiny. A more reasonable one would be noradrenaline in an adrenergic receptor, which makes like 5 or 6 hydrogen bonds, and it has a good affinity and very good specificity because these things are spatially arranged. Now, with odorants, which is what I've worked on, as Stuart mentioned, for a long time, the ligands get more slippery. The propionic acid, which is at the top here, makes 1 hydrogen bond with the olfactory receptor. But when you get to anesthetics, they simply just don't. And their interactions are extremely weak. And it's very hard to see what they're doing. But one thing that's become clear right from the start, in fact, due to an enormous amount of work by Meyer and Overton separately in the 1890s, is that if you plot the potency of anesthetics versus their ability to dissolve in a sort of oil, in olive oil, which is a mimic for both membranes and the parts of proteins that are hydrophobic. The relationship called the Meyer-Overton relationship is pretty good. You find that the more they dissolve in olive oil, the more potent they are. The graph on the left is a bit of a cheat. I think there's actually more variability than that, but we can still fit a decent line through it. It's hard. These experiments are hard to do, and they haven't been very much replicated. Now, what the Meyer-Overton graph really means is that if you have a log-log of a hyperbola, that gives you a straight line. And what it means is that essentially, what the anesthetic has to do is get to wherever it's going. And once that place is occupied by an anesthetic molecule, the animal will be anesthetized. So in some sense, leaving aside possible deviations from the Meyer-Overton, all anesthetics are equally potent, which is very strange. So you might say, well, where do anesthetics go? Now we have so many structures, and in fact, xenon is used as an aid to crystallography. Well, there's plenty of structures of proteins that have xenon binding sites. You can show xenon in them, and these are a few of them. And pretty clearly, the important binding site for anesthesia and consciousness is not going to be lysozyme or thermolysin or much less bacteriorhodopsin. So it's not very informative to have these structural data. There's 233 of those as we speak, and they're adding up because it's useful in crystallography. Now, where do general anesthetics go to? Rod Eichenhoff, who's done a lot of beautiful work in this field, has used anesthetics that are light sensitive and that grab onto the nearest protein when you shine the light. And he has listed them in order, in decreasing order of affinity for anesthetics. And you can see that these pink proteins are all mitochondrial. Now, in fairness to Stuart, and before he notices it and tells me off, tubulin is also in there, of course. There's lots of tubulin. It binds anesthetics, as we know. So the idea that there's some connection between anesthetics and mitochondria has, in fact, developed quite strongly in the last few years because, first of all, people who have mitochondrial disease respond weirdly to anesthetics, tend to be hypersensitive. And second, you can actually, in mice, you can show that one particular part of the electron transport chain of mitochondria, which is depicted very roughly here, the part on the left, which is complex 1, is necessary for anesthetics to work. If you replace parts of complex 1 with an unrelated complex of proteins from yeast, the animal still lives, but is no longer responsive to anesthetics. So this is very interesting. And if you look at anesthetics to the same scale as these proteins, this is kind of, it tells you where, at least you get a feel for the physical relationship between them and proteins. So electrons flow through this and protons flow through the water, and that's how mitochondria, which essentially power the cell, actually work. So we got interested in the electron part because xenon is famous, not least for this photograph, which was one of the early scanning tunneling microscopy pictures, where you have xenon atoms put on a gold surface, I think it was, and they moved them around until they were happy that it showed the company logo properly. And each of these little peaks indicates that a current is flowing through the xenon atom between the gold and the scanning tunneling tip. So we thought, well, it's interesting, maybe anesthetics somehow perturb electron transport or cause a leak or do some such thing. We weren't very clear about it. So what we did was we took some fruit flies, and I'll come back to why we're using fruit flies. Fruit flies are great for a whole bunch of reasons, chiefly because you can get lots of individuals that are exactly isogenic. They have the same DNA. They're clones, essentially, of each other. And you can put them in an electron spin resonance machine. And so the flies are in a tube, and you can pass any gas you like and hold them at any temperature. And they are in a magnetic field and a radiofrequency cavity. And the way it works, for those who never heard about electron spin resonance, is like this. You have an RF source, a cavity, a detector, and a magnet. And in the cavity, you have spins. And when the magnetic field increases, the separation between the up and down spin increases as well, until it reaches the right energy to absorb the photons that are coming from the RF source. And that's when you get a nice trace like that. This was discovered in 1947. And the other thing we have with Drosophila, which is absolutely amazing, is you have mutants. And in particular, you have anesthetic resistant mutants. And they were discovered using this wonderful machine on the left called an inebriometer by K.S. Krishnan. An inebriometer, what happens is you put flies at the top, you flood the thing with halothane, and the ones that are awake cling for dear life to these funnels and do not fall to the bottom. And so by essentially doing a sort of fly chromatography like this, you can select mutants. And Krishnan isolated 4 different mutants that are anesthetic resistant. And you can order them from the fly supply places and play with them. So what we did was we measured the spin inside flies, and this is a trace. So the pink trace is the noisy, is the trace as it comes out of the instrument, and if you low pass filter it, you get the black trace. And when you have the flies in nitrogen and then you add air, meaning 20% oxygen, the spin increases, and then it goes back to where it was with nitrogen. Now, this is due to respiration. You're increasing the number of electrons flying around in mitochondria. If you then put chloroform in nitrogen, you get an interesting signal, which really shouldn't be there because they're not actually respiring. There's no electrons flowing to oxygen. The ultimate point of arrival of electrons is oxygen, which is reduced to water. So clearly anesthetics do something, and then when you put the air as well as the chloroform, you get a smaller respiration signal, but nevertheless it's still there. So this is a very interesting picture. But what makes it really significant is that this anesthetic signal is pretty much reduced by, in 4 out of 5 mutants, is reduced significantly. So on the left, the brown column is the wild type flies, and there's considered the reference normal fly. And then the agar 11, 21, 52, 53 are mutants. And then there's a 211 where the difference is not significant. So, and this is what I'm plotting on the right is the amplitude of that anesthetic signal. So this is as direct as it gets, evidence that there's some connection between spin and possibly therefore mitochondria and the action of anesthetics. So we published this some years ago now, and to be honest, we didn't really know where to go from there, because you have this thing and you can show what I've just shown, but what's the next step? And the next step turned out to be a complete surprise, due to an amazing guy called Ron Naaman, who's really an outstandingly brilliant scientist, who discovered something that was completely out of the blue, namely that electrons in biological systems do not have random spins, that they're spin polarized. And the reason they're spin polarized is because when an electron travels through any solid which is chiral, and it can be a small chiral molecule or a protein or DNA indeed, or any number of things, as long as there's chirality, the spins become oriented in the direction of travel. This is pretty amazing. And this effect was discovered in 1999 and is now established. I've just been to a conference about this topic, and it's amazing. It's nearly 100% spin selectivity, spin polarization, even with a single layer of molecule. So if Ron Naaman, it's called chirally induced spin selectivity, CISS. If Ron Naaman's CIS mechanism applies, then all of biological electronics is essentially spintronics. And I believe this is very important for, among other things, neuroscience. So let me explain what we tried to do next. So my obsession has been, for a while, has been to figure out whether, in fact, electrons are spin polarized in living systems, because we suspect they are, but measuring it is a different story. So the idea, one of the things that we did was to try and detect emissions, radiofrequency emissions, not absorption as in the spectrometer, but emission. The reason you suspect this might happen is that under normal circumstances, spins are at thermal equilibrium. They're practically 50-50 spins in the up and down, because the energy caused by the magnetic field is really, really tiny compared to thermal energy. However, if the spins are actually polarized, let's say by flowing through a protein, then you're going to get a non-equilibrium situation, which must at some point must at some point cease, because the electrons will come out of the protein and will revert to equilibrium. And the idea was that when they reverted to equilibrium, you might get some radiofrequency emission if you had the flies in a magnetic field. So we knew what kind of frequency to look for, and indeed we found some interesting signals. And the reason this is interesting to neuroscience, of course, is that brain respiration uses 50 amps roughly of electron current all the time, night and day, whether you're awake or asleep. And in this reaction, which is the basic reduction reaction of oxygen to water, and under 1.4 volts roughly, which is the sort of chemical battery that powers the whole thing, and that's 70 watts, which is pretty impressive. And a fruit fly, 1 microwatt, but we have 30 of them in the tube. So we built a special machine to detect radiofrequency emissions from flies. And we did detect some, but they were hard to get, in the sense that you could do a lot of experiments and see nothing. And then, one experiment would give you spectacular bursts of radiofrequency emission. And we got so cheesed off about this that in the end we decided to be brutal. And what we did was we used mutant flies that were genetically altered, that have a channel in every neuron which is sensitive to temperature, so that when the temperature rises above 30, 31, the neurons start firing like crazy. So essentially they have seizures. And here you can see, I don't know if the videos will show properly, but here we go. You can see on the right, the rightmost two are having seizures because they've been just immersed. The container's been immersed in 35-degree water. So we did this with the flies in the resonator I've shown you. And what happens is you put them in cold at 4 centigrade, and then they heat up during the experiment. And at some point, you can see that radiofrequency emissions are shown in red. You can see that they start and they become really intense. And at the end of this experiment, the flies were alive. Now, if you do the Fourier analysis of the frequencies going on there, our bandwidth is 50 hertz maximum with those experiments because we use a Luckian amplifier. You can see that there's activity at all frequencies when the bursts are happening. And another experiment, similar experiment, you can see that they have essentially clonic convulsions, and the bursts are separated by a relatively quiescent passage. The inset is one of those bursts, showing there's a lot of substructure there. Now, in this experiment, the flies were dead at the end. We waited too long. They were 34. They didn't survive. So this is all very interesting and indicative that something indeed may be going on. But I was always mindful that there might be some simpler way to approach this, and in particular, that all of this didn't tell us what anesthetics actually did. And so what happened was last year in June, I went to a congress in Crete on quantum biology, as it happens, and Ron Naaman, the discoverer of CIS, was there. We had a beer, and I started telling him about anesthesia, and he said, "Oh, this is very interesting. What's anesthetic?" And to show off and to point out to him that structure-activity relations were difficult to figure out, I said to him, "Xenon," and he said, the magnificent Israeli accent, "But then I understand." He said, "It is spin-orbit coupling. Xenon will scramble spins." And so there and then we decided to do some experiments together. And the idea is that the oxygen reduction reaction, which is central to mitochondria, is a spin-sensitive reaction because oxygen is what's called a ground state triplet. It has two unpaired spins pointing in the same direction rather than the usual one up, one down. So oxygen is paramagnetic. And Faraday showed that, I think, in the 1820s by blowing bubbles full of oxygen and attracting them to a magnet in front of an audience of children. So what's the experiment? Well, the oxygen reduction reaction is going to be spin dependent for a very simple reason. If you have an oxygen molecule with the two spins aligned, and you feed it two aligned spins in the opposite direction, it's going to like it because it's easy. The two spins you're adding, because they're polarized, will add easily onto the triplet of the oxygen molecule, and they will give you an oxide anion, and then the reaction proceeds from there. However, if you give unpolarized spins, and they're shown as the blue in the bottom here, well, the reaction proceeds more with great difficulty because you can't add those spins as they are to the oxygen molecule. It's forbidden. So the idea was to check this mechanism using electrochemical technique and see whether anesthetics made a difference, as Ron predicted. So the way this is done is you have a ferromagnetic electrode, and it's made of titanium with a layer of nickel and then gold to protect the nickel from oxidation. And you have a permanent magnet. And if the permanent magnet is some distance away at a very low field at the electrode, the spins come out about 50-50, polarized in opposite directions. If you bring the magnet right up to the electrode, however, the spins come out polarized. I've exaggerated the amount of polarization here. I think it's roughly 50%, but more than normal. So when you do the experiment, you have a working electrode, the one I've just described. You have your reference, you have an oxygen cylinder, goes through a anesthetic vessel. And here's what happens. You move the voltage from 0 to more negative values to reduce the oxygen. Now look at the O2 curve, the red curves. The solid red curve is with a magnet, and the dotted red curve is without the magnet. So what you can see is that the presence of a magnet that is not in contact with the solution, I think, just behind the electrode, makes quite a difference to the oxygen reduction current. But the amazing thing is when you put chloroform on, first of all, the voltage peak of the oxygen reduction shifts, showing that the reaction is actually changing. And secondly, the effect of the magnet becomes less pronounced. And you can do this, that's with chloroform. You can do this with halothane, where you get a spectacular result, where practically the oxygen refuses to be reduced, and magnetic field has no effect. That's on the left. And on the right, again, you have the effect of magnetic field on the oxygen reduction. And with nitrous oxide, you practically don't get any change in current. So this is really quite remarkable, and it was kind of interesting because we tried to publish. Ron was absolutely dead keen on sending this to Nature or eLife or some really big journal, whatever, and the biologists totally didn't get it. They said, "Well, who cares?" Basically, you know. And so in the end, we published it in the Journal of Physical Chemistry Letters, which I'm delighted about. But because the physical chemists thought this was really interesting, because they're interested in cis and electrochemistry and so on. So to sum up, it's now pretty clear that inhalational anesthetics in general mess with mitochondrial electron transport. It's also pretty clear that they are capable of scrambling a fundamental property of electrons moving around in chiral phases, namely spin polarization, at least in vitro. But, of course, the big question is, what's going on in vivo, and how can we prove it? And that's what we're working on at the moment. And in particular, we're trying to understand whether the series, you can see on the left in complex 1, there's a series of iron-sulfur groups where the electrons jump from one to the other. We're trying to figure out whether we can do measurements to show that the spins are actually polarized in that group of proteins. And all this work was done by myself and Ekin Daplan, my graduate student at the University of Buckingham, Alexandros Gaetanidis at the Alexander Fleming Institute near Athens in Greece, where I used to work until I came to the UK, and Antonello Soggiu, who is a RF and magnetics wizard from L'Aquila near Rome in Italy. And it was funded by Stavros Niarchos Foundation, DARPA, who've been very generous, and Ionis, a San Diego-based pharmaceutical company who has a Blue Skies Research support program. Thank you for your time and patience, and that's all I have to say.
Good. Thank you.
Luca, thank you very much. Do you have any? I have a question. Are you saying does this mitochondrial effect have any effect on? Are you saying that's what inhibits consciousness?
Well, I mean, it's an enormous gap between, as I'm sure you'd appreciate, between what we have and saying anything about consciousness. But the minimal hypothesis, which I'm not happy with, but passes the laugh test, should we say, is that anesthetics will inhibit respiration to a limited amount. In other words, the spin scrambling will screw up, let's say, 30 to 40% of the efficiency of mitochondria wherever they are in the brain. So if this slight hypoxia, so to speak, is sufficient to disable synaptic function, as many people have proposed, that synapses have, almost every synapse has a mitochondrion in it to supply it with power. So if indeed anesthesia is, as Bruno van Swinderen has said, is a synaptic power failure, then we have a good reason for it. Personally, since I'm talking to an open-minded audience, and Stuart in particular, I don't think that's the answer. I think this is an insight into something that we don't understand, okay? that may have to do with spin, and we have to do with some quantum aspect of consciousness that we don't fully understand yet. But the problem is, if you say that to an audience of pharmacologists, they call an ambulance to take you away.
Santosh.
Hi, it was a nice talk. You left out complex 2, and I'm wondering why. Because we have, in my other work, I have used a magnetic stimulation device and shown that succinate dehydrogenase activity is reduced by magnetic rotating magnets. So I was wondering.
So yes, my apologies for leaving out complex 2. I'm busy learning mitochondrial physiology as I go along, and yes, indeed, and in fact, complex 2 probably will be affected much in the same way insofar as electrons are flowing through it. The only reason I focused more on complex 1 is because the mutants of complex 1 that affect the anesthetic response are really well characterized.
Okay. Thank you.
One more.
Thanks, Michael. Are you familiar with Ron Naumann's other story about SDCR, charge-dependent, spirally dependent charge reorganization?
Yes, I'm familiar with it. In fact, we published a paper in Physical Chemistry Letters last year showing that it could explain a long-standing but mysterious effect on viscosity. The spin-dependent charge reorganization is perhaps the most difficult part of Ron Naumann's work to actually confirm experimentally. But as I said, I've just been to a Congress, and I think a number of people are working on that. It's absolutely fascinating, the idea that fluctuations in charge within a molecule can cause spin redistribution.
Well, I've done a paper here that shows that that when you connect to different chiralities, if they're unalike, they hook up. If they're alike, they don't. So all the amino acids in tubulin, if there's left and right in there, they have a trouble hooking up and are faulty, and that is a major cause of all the mental and health brain diseases as we see today because of this repulsion of the left and right when you try and hook two up. So it's a
yes, if you have, so my understanding is that the phenomenon you're talking about depends crucially on a large polarizability. So the larger the polarizability, the bigger the effect will be. And it's not clear how high you can go, but I wouldn't be surprised if it did.
Luca, are you familiar with the study from China a couple years ago about isotopes of xenon that had a 1/2 spin, and they were less potent because they thought the half spin promoted consciousness and inhibited the anesthetic effect? But that's nuclear spin.
I'm not familiar with it, and to be perfectly honest, I would want that thing to be replicated a couple times before I started losing sleep over it, simply because it was a peculiar paper in the sense that they used 80% of perfectly normal anesthetic and then added the isotope simply because they had small quantities of these weird isotopes of xenon.
Yeah, that's
And
good.
so,
The macro of xenon is too weak. That's why they had to do that.
right.
So otherwise, they were hypoxic.
But I guess my feeling is that is almost by definition the hardest experiment in the world to replicate, because getting a hold of those isotopes of xenon, I mean, normal xenon is bad enough right now, but those dudes are really impossible to get. So until someone replicates that, I'm just going to keep my powder dry.
Fair enough. Well, Luca, thank you for staying up late for us. We appreciate it very much.
It's been a pleasure.
Great talk.
It's been a pleasure.
And let's
Thank
give him
you.
a round of applause. Thank you.